And let's talk about Chapter 2, Senses and Perception. You can think of your sense or organs as the brain's windows on the external world. The world itself has no actual images, sounds, tastes, and smells. Instead, you are surrounded by different types of energy and molecules that must be translated into perceptions or sensations. For this extraordinary transformation to work, your sense organs turn stimuli such as light waves or food molecules into electrical signals through the process of transduction. These electrical messages are then carried through a network of cells and fibers to specialized areas of your brain where they are processed and integrated into a seamless perception of your surroundings. And first, vision. Vision is one of your most complicated senses, involving many pro- processes that work simultaneously, enabling you to see what is happening around you. It is no surprise then that the visual system involves about 30% of human cerebral cortex, more than any other sense does. Vision has been studied intensively, and we now know more about it than any other sensory system. Knowledge of how light and energy is converted into electrical signals comes primarily from studies of fruit flies and mice. Higher level visual processing has mostly been studied in monkeys and cats. In many ways, seeing with your eye is similar to taking pictures with an old-fashioned camera. Light passes through the cornea and enters the eye through the pupil. The iris regulates how much light enters by changing the size of the pupil. The lens regulates how much light enters by changing the size of the pupil. The lens The iris regulates how much light enters by changing the size of the pupil. The lens then bends the light so that it focuses on the inner surface on your eyeball, on a sheet of cells called the retina. The rigid cornea does the initial focusing, but the lens can thicken or flatten to bring near or far objects into better focus on the retina. Much like a camera capturing images on film, visual input is mapped directly onto the retina as a two-dimensional reserved image. Objects to the right projected images onto the the left side on the retina and vice versa. Objects above are imaged at the lower parts and vice versa. After processing by specialized cells in several layers of the retina, signals travel via the optic nerves to other parts of your brain and undergo further integration and interpretation. And this picture shows how visual information is transmitted. And the three-layered retina. The retina is home to three types of neurons: photoreceptors, interneurons, and ganglion cells, which are organized into several layers. These cells communicate extensively with each other before sending information along to the brain. First, the photoreceptors. It's the light sensitive, and it contains rods and cones, are located in the most peripheral layers of the retina. This means that after entering through the cornea and lens, light travels through the ganglion cells and interneurons before it reaches the photoreceptors, and then ganglion cells and interneurons. and they do not respond directly to light, but they process and relay information from the photoreceptors. The axons of ganglia cells exist, the retina together, forming the optic nerve. And this picture above shows a three-layered retina. There are outer layer, middle layer, and inner layer. And second, hearing. Hearing is one of your most important senses. Alerting you to an approaching car and telling you where it's coming from long before it comes into sight. Hearing is also central to social interactions. It allows you to communicate with others by processing and interpreting complex messages in the form of speech sounds. Like the visual system, your hearing system picks up several qualities of the signals it detects, such as a sound's pitch, loudness, duration, and location. Your auditory system analyzes complex sounds, breaking them into separate components or frequencies. As a result, you can follow particular voice in a conversation or instrument as you listen to music. And sound in the form of air pressure waves reaches the pinnae of your ears, where the waves are funneled into each air canal to reach the eardrum. The eardrum vibrates in response to these changes in air pressure, sending these vibrations to three tiny sound amplifying bones in the middle ear, the malleus, incus, and stapes. The last bone in the chain acts like a tiny piston, pushing on the oval window, a membrane that separates the air-filled middle ear from the fluid-filled snail shell-shaped cochlea of the inner ear. The oval window converts the mechanical vibrations of the stapes into pressure waves in the fluid of the cochlea, where they are transduced into electrical signals by specialized receptor cells called hair cells. And this is the picture shows the process of hearing. And how the pressure wave transformed to electrical signals. And an elastic membrane called the basilar membrane runs along the inside of the cochlea like a window ramp, spiraling from the outer coil near the oval window to the innermost coil. The basilar membrane is tuned along its length to different frequencies. When fluid inside the cochlea ripples, the membrane moves, vibrating to higher pitched sounds near the oval window and to lower pitched sounds in the center. Rows of the small sensory hair cells are located on top of the vibrating basilar membrane. When the membrane moves up and down, microscopic hair-like stereocilia extending from the hair cells band against an overlying structure called the tectorial membrane. This banding opens small channels on the stereocilia that allow ions in the surrounding fluid to rush in, converting the physical movement into an electrochemical signal. Hair cells stimulated in this way then exit the auditory nerve, which sends its electrical signals onto the brainstem. The next stop for sound processing is the thalamus, the brain's relay station for incoming sensory information, which then sends the information into the auditory part of the cerebral cortex. Several thousand hair cells are positioned along the length of the basilar membrane. Each hair cells respond most strongly to just a narrow range of sound frequencies, depending on how far along the cochlea it is located. Thus, each nerve fiber connecting with the higher cells is tuned to very specific frequencies and carries this information into the brain. And third, taste and smell. The senses of taste and smell are closely linked and help you navigate the chemical world. Just as sound is the perception of air pressure waves and sight is the perception of light, smell and taste are your perceptions of tiny molecules in the air and in your food. Both of these senses contribute to how food tastes, and both are important to survival because they enable people to detect hazardous substances that may inhale or ingest. The cells processing taste and smell are exposed to the outside environment, leaving them vulnerable to damage. Because of this, taste receptor cells regularly regenerate, as do olfactory receptor neurons. In fact, olfactory neurons are the only sensory neurons that are continually replaced throughout our lives. And first about the taste. Our ability to taste food depends on the molecules set free when we chew or drink. These molecules are detected by taste cells within taste buds, located on the tongue and along the roof and back of the mouth. We have between 5,000 and 10,000 taste buds, but start to lose them around age 50. Each taste bud consists of 50 to 100 sensory cells that are receptive to one of at least five basic taste qualities: sweet, sour, salty, bitter, and umami. Contrary to common belief, all tastes are detected across the tongue and are not limited to specific regions. When taste receptor cells are stimulated, they send signals through three cranial nerves: the facial, glossopharyngeal, and vagus nerves, to taste regions in the brainstem. The impulses are then routed through the thalamus to the gustatory cortex in the frontal lobe and insula, where specific taste perceptions are identified. And second, the smell. Odors enter the nose on the air currents and band to specialized olfactory cells on a small patch of mucous membrane high inside the nasal cavity. Axons of these sensory neurons enter the two olfactory bubbles after crossing through tiny holes in the skull. From there, the information travels to the olfactory cortex. Smell is the only sensory system that sends sensory information directly to the cerebral cortex without first passing through the thalamus. We have around 1,000 different types of olfactory cells, but can identify about 20 times as many smells. The tips of olfactory cells are equipped with several hair-like cilia that are receptive to a number of different orders molecules, and many cells respond to the same molecules. A specific smell will therefore stimulate a unique combination of olfactory cells, creating a distinct activity pattern. This signature pattern of activity is then transmitted to the olfactory bulb and onto the primary olfactory cortex, located on the anterior surface of the temporal lobe. Olfactory information then passes to nearby brain areas, where odor and taste information are mixed, creating the perception of flavor. Recent research suggests that people can identify odors as quickly as 110 milliseconds after their first sniff. Interesting, Interestingly, the size of the olfactory bubbles and the way neurons are organized can change over time. As mentioned above, the olfactory bubbles in rodents and primates, including human, are one of the few brain regions able to generate new neurons throughout life. And there are two pictures. The first pictures shows the taste bulbs transform information about taste and send them to the brain to be processed into your favorite flavors. In this image of a mouse tongue, the axons that connected to these receptors are highlighted in red. And the second picture shows the olfactory bulb is a structure in the forebrain responsible for processing smell information. This series of images shows the olfactory bubbles from a zebrafish at three stages of development. And there are some interesting things about taste and smell, about combining taste and smell. Taste and smell are separate senses with their own receptor organs, yet we notice their close relationship when our nose is stuffed up by a cold and everything we eat tastes bland. It seems like our sense of taste no longer works, but the actual problem is that we detect only the taste, not taste and smell combined. Taste sense itself is rather crude, distinguishing only five basic taste qualities, but our sense of smell adds great complexity to the flavors we perceive. Human studies have shown that taste perception are particularly enhanced when people are exposed to matching combinations of familiar tastes and smells. For example, sugar tastes sweeter when combined with the smell of strawberries than when paired with the smell of peanuts, butter, or no odor at all. Taste and smell information appear to converge in several central regions of the brain. There are also neurons in the inferior frontal lobe that respond selectively to specific taste and smell combinations. Some of our sensitive to taste and smell is lost as we age, most likely because damaged receptors and sensory neurons are no longer replaced by new ones. Current research is getting closer to understanding how stem cells give rise to the neurons that mediate smell or taste. With this knowledge, stem cell therapies might one day be used to restore taste or smell to those who have lost it. And this is all the things about chapter 2. Thank you.